Hello friends, welcome to my course on microbial biotechnology. Today we are starting module number 7, which will deal with microbes in agriculture, biofertilizers, microbial pesticides, and finally the concept of integrated pest management. So let us start with overall the role of microbes in agriculture. So we have divided this lecture into various sections. We start with microbes, frenemies of agriculture. They are both friends and enemies of agriculture at the same time. Then we'll discuss about the microbiome in soil, then microbial groups in soil and their interactions, then microbes in soil fertility and crop production, and plant growth promotion and phytohormones. We'll tell about the sections 2 and 3 as we go to them in the subsequent slides. Before we begin, let us make some comparison between the health of an individual and health of the ecosystem. So all of you know that our gut is full of microbes, and so is our skin. So now we have two types of microbes which inhabit us in the gut and as well as in our skin. One are beneficial and others are harmful. So the harmful ones cause us diseases like upset stomach, then some kind of skin infections and other disease conditions. But there are many beneficial gut microflora which helps us in our well-being. Similarly, if you look into the ecosystem, we have diverse groups of microbes inhabiting every niche of these ecosystems. Some of them are beneficial and some of them are harmful. So the beneficial ones helps the ecosystem by recycling nutrients. We have learned about this in earlier slides, as well as maintaining the homeostasis of the ecosystem. Now the health and well-being of an individual is the balance between the good and the bad microbes, and many of these good microbes actually help us in controlling the bad microbes, and it is also other way around. Sometimes the bad microbes dominates over the good microbes depending on the ecological balance, whether it is a micro environment or the macro environment. And in a unhealthy ecosystem where there is some kind of unhealthy human interventions, the bad microbes may dominate and the good microbes may altogether disappear, further deteriorating the environment. So similar would be the case if we do not maintain good hygienic practices or food habits, or even when we consume a lot of antibiotics, a similar condition may happen to us in the individual level. Now you already have some idea about the soil being inhibited by both beneficial and harmful microbes. So these whole soil environment contains a complex ecosystem of microorganisms, and together we call them as the soil microbiome. In the way we call the gut flora as the gut microbiome. The science of soil microbiology explores how these microorganisms interact with each other and the environment, shaping soil conditions and creating a habitat for diverse life forms. So these are the most lowest forms of organisms which inhabit and actually make the soil amenable for other higher organisms like plants to survive on them. And these interactions influence the soil properties, the quality of the soil, the depth of the soil structure, and even the texture. So what the microbes do in the soil, what are their functions? There are various functions. Number one, they act as decomposers, so helping in the decompositions of organic matter, and then some of them produce phytohormones or microbial phytohormones, and these phytohormones help the plants in their growth and development. And they also do nutrient recycling, where they decompose the organic matter into simple substances and nutrients which is taken up by the plants, and then they sometimes develop, some of these microbes develop symbiotic relationships, and that where both the organisms are benefited and are of mutual interest. And then they also help in soil structure and aggregation. For example, the sand may be aggregated with the help of certain microbes like fungi, bacteria, and these may be bound to the clay particles, or two sand particles may be bound to one another, thereby helping the aggregation and solidification of these soil structure. And then many microbes help in regulating the salinity of soil, thereby making the soil conducive for the growth of plants. The microbes also helps in disease suppression, and we can use many of these as biocontrol agents against certain pathogens. So, overall we can see that without microbes in the soil would actually become very, very inhabitable or non-friendly to higher organisms like plants. So, what are the different groups of microbes in soil? So, we may have bacteria, actinomycetes, fungi, soil algae, protozoa, and bacteriophages. You can see the various examples of these like Agrobacterium, Actinomyces, then Aspergillus fungi, then Anabaena algae, and then you have nematodes, and then Colpoda, and then of course we have bacteriophages, which are not microbes. They are also present in soil and they contribute in the ecological balance. So, when it comes to bacteria, they are the most dominant group of microorganisms in soil, and they are involved in organic matter decomposition, biogeochemical cycling of elements, and antibiotic production. Actinomycetes are capable of degrading complex organic substances contributing to soil fertility, and they are notable for antibiotic production. And fungi are the second most dominant organisms after bacteria. They prevail in acidic soils, but can thrive in all types of soils influenced by environmental factors and organic matter content. Soil algae are ubiquitous, visible as green scum where moisture and sunlight are present. Cyanophyceae fix nitrogen, and these are influenced by environmental factors as in the case of other microbes. The protozoa are unicellular organisms with cysts in their life cycle. They prefer specific bacterial species for nutrition, abundant in upper soil layer depending on the bacterial population. How do microbes interact with one another? One type of interaction is the beneficial one, while the other is the harmful interaction. In beneficial interaction, they form symbiotic relationship, which is called as symbiosis or mutualism. Here microbe A and B, or organism A and B, benefit each other, as you can see by the two directional arrows here. And in certain cases this benefit is only one directional. So, A benefits B, but B do not benefit A. Such a relationship or interaction is known as commensalism. We have, for example, microbiota that thrive on the skin and in the gut. They help us in many ways. Then there are harmful interactions like amensalism, where A harms B. Pseudomonas aeruginosa is antagonistic towards Aspergillus terras, and there is parasitism, where A harms B, but B actually benefits A. So, for example, malaria caused by Plasmodium, a protozoan transmitted to humans by Anopheles mosquito. So, we have to observe this parasitism and these symbiosis a little bit keenly, where symbiosis is a case where both the organisms are benefited, but in parasitism one organism is benefited while the other actually harms it. So, what are the different kinds of advantages having soil microbes? So, they basically play a big role in soil fertility and thereby in the crop production. So, they enhance the soil health through various mechanisms and support its overall vitality. For example, they create soil structure, which we have just studied. Then they recycle waste and detoxification of harmful compounds. They serve as the growth medium for plants, and then provides raw materials, help maintain soil moisture, and they also act as carbon storage and helps in the nutrient recycling, and many times control pests, weeds and pathogens, and this is very important for us in this particular lecture. Let us now look into plant growth promoting rhizobacteria, which we call as PGPR, that enhances plant growth, particularly in pathogen free conditions. So, they act as eco-friendly biocontrol agents by competing for resources, excluding pathogens and producing antifungal metabolites like hydrogen cyanide, phenazenes and pyrrolnitrine. They also induce systemic resistance in plants through jasmonate and ethylene signaling, bolstering plant defenses. Key bacterial components such as lipopolysaccharides, flagella, siderophores and volatiles play crucial roles in ISR, promoting plant health and resilience. So, you can see here that PGPR can help directly by carrying out nitrogen fixation, releasing phytohormones, then ACC-deaminase and water soluble B vitamins, P and K solubilization, mineral solubilization, then siderophore production. And they also help by indirect mechanisms where they generate ISR responses, prevention of plant diseases, and also in the control of pathogens. Let us now have a small discussion on phytohormones produced by microbes, which we call as microbial phytohormones. So, they plant physiological functions like growth, development and responses to environmental stimuli are regulated by various phytohormones such as abscisic acid, salicylic acid, cytokinins, gibberellins, brasinosteroids, auxins, jasmonic acid and ethylene. These hormones interact cooperatively or antagonistically. So, plant growth promoting bacteria PGBB can synthesize or degrade many phytohormones and produce volatile organic compounds which enhance plant growth and influence phytohormones expression. So, some of the phytohormone structures like cytokinin, ethylene, then gibberellin, auxin and abscisic acids, which are also produced by plant and many other organisms. Azospirillum brasilense and Bacillus species produce these cytokinins, which promotes cell division and overall plant growth, especially in the shoot and the root portion. Rhizobacteria like Azospirillum and Bacillus produce indole acetic acid that helps root growth grow longer and stronger, improving the plant's ability to absorb water and nutrients. Bacillus licheniformis and Bacillus pumilus produce gibberellins, which help plant grow taller, develop more balanced branches and form healthy roots. So, here we can see plant phytohormones, which some of which act as growth promoters like auxins, gibberellin, cytokinin, salicylic acid, ACC-deaminase, and then we have growth inhibitors, ethylene, abscisic acid and jasmonate. And there are bacterial substances that affect the hormonal regulation of plant by producing growth promoters like auxins and then gibberellins and then cytokinins, salicylic acids, ACC and also volatile organic compounds, and it also produces growth inhibitors like ethylene and abscisic acid. The role of plant microbiome phytohormones in different plant growth activities. So, plant microbiomes producing, for example, gibberellic acid, auxin and brasinosteroid. So, they help in different ways. Gibberellic acid, for example, will help in reproduction, flowering and then lateral organ formation. Auxin will help in reproduction and then lateral organ formation and cell wall modification. Similarly, gibberellic acid may also help play a role in germination, leaf serration and then root growth. Auxins also helps in root growth. So these are, you can see many roles played by the phytohormones released by microbes. Overall, microbial phytohormones enhance plant stress tolerance through various mechanisms as well. So root associated microbes produce hormones which enhance the plant's antioxidant defenses, reducing oxidative stress. It improves photosynthesis and stabilizes cell membranes, promotes cell division and regulates stomatal function, enhance root growth and increase water and nutrient uptake. So when a plant is under abiotic stress like heat, drought, heavy metal salt and even cold, so it will have excess ROS accumulation, the membrane dysfunctioning, nutritional disorder, hormonal imbalance, reduced photosynthesis and protein synthesis. And the phytohormones released by microbes will help in the improved germination, upregulated antioxidant system, then reduce ROS, which was increased due to this stress, then induction of system acquired resistance and also assistance in the cell signaling. So we see that microbial phytohormones play a very crucial role in enhancing stress tolerance in plants. So this is a list of certain bacteria which produce phytohormones, and it also shows the phytohormones they produce, and these overall mitigate abiotic stress in certain selected plants which are listed over here. For example, these Pseudomonas species and Bacillus species, they produce indole acetic acid in certain plants like Sula carnosa, and then we have Bacillus laciniformis which also produce IAA in Triticum aestivum, and then we have others and a host of other microorganisms which produce indole acetic acid from here to here, and in diverse plant systems like Cucumis and then Hordeum, Acacia, Giamase and so on. Then we have also some other organisms like Achromobacter, which produces here acid, which is helpful in providing Helianthus anus the stress tolerance. Then we have Micrococcus luteus and several others like Bacillus subtilis, which produces cytokinin, and then we have Azospirillum which produces gibberellic acid, and then others which produce abscisic acid, and there are some organisms which produce host of phytohormones together, whether it is indole acetic acid or gibberellic acid or abscisic acid. Let us now discuss about an important topic, climate resilience, and how microbes help in overcoming, adapting to climate resilience challenges. So let us start with the role of microbes in stress response against climate resistance. We will also go for discussing about bioremediation and certain biochemical cycling of nutrients in this particular section. So we know that currently the rising global temperatures and increased extreme weather conditions and events are getting driven by climate change and making adaptation crucial to protect agro ecosystems and forest ecosystems and ocean ecosystems, and also to safeguard societies and nations. Agriculture is largely affected by climate change already. It disrupts plant homeostasis and causes stress through factors like drought, salinity, heat and flooding. Microbes assist plants in managing abiotic stress like drought and extreme temperatures. For example, we have here say the effect of climate change, which will induce drought, cause high salinity, and it results in a lot of heat and as well as flooding. Now, under these different stress conditions, the plants will have different kind of stresses like osmotic stress and oxidative stress, as you can see in this diagram. Whenever there is a low water content, there will be osmotic stress because the soil will become very hard. And then there is also ionic stress due to high salinity, for example, high sodium concentration, and all these ionic stress and osmotic stress would lead to oxidative stress. And due to flooding, there is a low oxygen concentration, and then there is a fermentation, phytotoxic compounds are produced. These are also lead to the oxidative stress, and due to heat, there is an altered membrane and photosynthesis, which also adds to this oxidative stress. And in the earlier slide, we have discussed how oxidative stress is harmful and how plant microbial phytohormones help in overcoming these oxidative stress. So, there are numerous mechanisms that help in promoting growth in stress conditions induced by climate change. Two of them are reduction of ethylene levels in plants and countering the effects of high salinity in plants. So, we know that high salinity causes ionic stress leading to oxidative stress, and we will try to discuss how these help. So, Pseudomonas species support plant growth under stress by producing hormones, emitting beneficial compounds, reinforcing root barriers, reducing oxidative stress, and secreting exopolysaccharides. These actions mitigate stress effects and enhance growth, ultimately improving yields despite adverse conditions. So, this is the Pseudomonas species. So, it is carrying out plant hormone biosynthesis and modulation, releasing volatile organic compounds and also producing exopolysaccharides, and it is strengthening apoplastic barriers and also helping in the decreased oxidative stress, which helps in adapting against various climate change things like heat, water, flooding and so on. So, these leads to improved decreased effects of climate related stress and improved plant growth under climate related stresses. So, the versatility of Pseudomonas strains offers valuable opportunities for developing bioinoculants to combat climate related diseases. So, we know the beneficial effects of Pseudomonas, so many, and then if we apply these into crops, we can take advantage of all these beneficial properties. So, let us discuss the reduction of ethylene levels in plants. For example, Pseudomonas strains promote plant growth under stress by producing the enzyme ACC deaminase. So, ACDS, what we call in short, reduces levels of 1-amino cyclopropane, 1-carboxylate, which is the ACC. It is a precursor to the stress related hormone ethylene by breaking it down into ammonium and alpha ketobutyrate. So, this is ACC compound. So, this particular enzyme ACC oxidase produced by Pseudomonas will reduce plant growth under stress, because it won't help in the production of ethylene, because it breaks it down into ammonium and alpha ketoglutarate, as you can see here. So, in the root, this product is available, compound is available, which is taken by the bacteria, and here it is converted into compounds, thereby stopping the production of ethylene. So, there is ethylene regulation. The reduction in ACC lowers ethylene levels in plants, alleviating stress effects such as premature leaf drop and reduced root development. Consequently, plants can maintain physiological functions and continue to grow despite adverse conditions. Now, there is a mutual benefit over here. The breakdown products of ACC, ammonium and alpha ketoglutarate, serve as usable nitrogen and carbon source for Pseudomonas, enhancing bacterial growth and survival in the rhizosphere. So, these ultimately serve as the nitrogen and carbon source in the bacteria. So, they have a mutualistic benefit over here. Second is the countering the effects of high salinity in plants. So, we can see here a figure A, root apoplastic barrier without Pseudomonas inoculation, and in B we can see root apoplastic barriers reinforced by Pseudomonas inoculation under high soil salinity conditions. So, for example, in Pseudomonas mandeli strain IBKI14 improves soil tolerance in wheat and pea by enhancing suberin and lignin deposition in xylem cell walls, suberin lamellae and Casparian strips. So, you can see here the suberin lamellae here, and then there is the Casparian strips. So, when there is no inoculation, there is high apoplastic transport of ions and high apoplastic transport of water. When there is inoculation by these particular Pseudomonas strain, there is low apoplastic transport of ions and high symplastic transport of water. Let us now discuss another important topic, the bioremediation. Microbes can remediate soil contaminated with pollutants and chemicals, contributing to soil health restoration. So, we call bioremediation as an eco-friendly strategy. So, you can see here bacteria non-flagellated and bacteria flagellated. This is the flagellated one and this is the non-flagellated one, and we can see fungal hyphae over here, and then we can also see the biosurfactants. Here you can see these are released biosurfactants. Okay. Now, how it helps? There is natural attenuation, less cell count and then slow degradation. Native microbes degrade pesticides at a very slower rate. So, then we do bio augmentation, where we have enriched degraders and engineered degraders. So, introducing enriched or genetically engineered microbes to accelerate pesticide degradation in the environment. Although there is a lot of reservation regarding introducing genetically engineered microbes, which we will discuss in later modules. Then there is biostimulation, which increases growth and increases bioavailability. This involves the addition of nutrients and biochemicals to boost activity of existing microorganisms. So, here we have naturally occurring microbes. So, in biostimulation we will add up nutrients and chemicals to boost the activity of existing microorganisms. In bioaugmentation, we will be introducing genetically enriched microbes instead of introducing nutrients. One of the most important role of microbes in soil is the cycling of nutrients, which we call as biogeochemical cycling. This involves the recycling of essential elements such as carbon, nitrogen, phosphorus and sulfur through biological activities by plants, animals, microorganisms and also the geological processes. So, for example, here this is a tree, the leaves are dropped, and also sometimes the tree is dead or it falls down due to storm or wind conditions, and also animals die. So, we have the dead plant and animal matter. So, these are decomposed or break down into organic matter in the soil by various organisms, and then these release minerals and other nutrients into the soil, helps in the plant growth. That is one cycle, and also the rock present below the soil gets broken down, and in these entire decomposition and breaking down of both the organic matter as well as mineral rocks, microbes play a big role, and this helps in maintaining the ecosystems where both plants and animals survives. So, microbes are crucial in these cycles. It transforms and recycles nutrients which are then released back into the environment. Such cycling maintains ecosystem health, supports plant growth and animal growth, and ensures a balanced nutrient supply, contributing to ecological balance, agricultural productivity and environmental protection. So, what are the various soil microorganisms which take part in nutrient cycling and maintaining soil fertility? So, for sustainable agricultural productivity, we need good and healthy soil which is fertile, and this is significantly influenced by soil microorganisms like bacteria, fungi, archaea and protozoa. So, these microbes are crucial for biogeochemical cycling of essential nutrients like nitrogen, phosphorus, sulfur as well as others, as we will be discussing in this module and also in another module. So, this enhances nutrient availability, supports plant growth and its sustainable land management. Microbial transformation and mobilization of nutrients for plant accessibility. So, one important aspect is nitrogen fixation, and then the nitrification and also denitrification in the case of nitrogen recycling. So, in nitrogen fixation, soil bacteria like Rhizobium species and free living species like Azotobacter convert atmospheric nitrogen into ammonia, making it accessible to plants. In nitrification, ammonia is oxidized here you can see to nitrate and then nitrite and then to nitrate by bacteria, with nitrate being more readily absorbed by the plants. In the denitrification process, denitrifying bacteria convert nitrates and nitrites into nitrogen gas or nitrous oxide here, completing the nitrogen cycle. So, if you start from nitrogen, we go through nitrogen fixation, nitrification, then finally denitrification, and we are back to where we started. And in this entire process, the nitrogen is recycled between organisms and different environmental niches. Let us look into the recycling of phosphorus. So, we have mineralization in the case of phosphorus. Microorganisms decompose the organic matter to release inorganic phosphate from organic phosphorus. So, here you can see this is inorganic phosphorus and microorganisms act on it. Then by the process of mineralization, it sends it into solution form, and then the reverse is immobilization, where microbes temporarily store phosphorus in their biomass here, which is later released back into the soil upon microbial death and decomposition. So, solubilization is held by certain bacteria and fungi that dissolves insoluble phosphorus compounds, making them available to plants. So, ultimately phosphorus which is absorbed in hematite and other minerals, and then here they will actually come to this solution form, and also those from the organic matter will come to the solution form, and these become available to the plants. And of course, when we apply fertilizers artificially, that also gets solubilized, and finally it enters the plant system. And the remaining which cannot be absorbed or even before absorption by plants will be lost by erosion or leaching, but they will end up somewhere in the environmental reaches, and then it will enter this cycle, and then it will again become available to some other plants. The next important thing is sulfur recycling. So, we have two processes over here, the oxic processes and the anoxic processes. So, in sulfur oxidation, sulfur oxidizing bacteria convert sulfur and hydrogen sulfide to sulfate, which plants can absorb easily. In the sulfur reduction cycle, sulfate reducing bacteria reduce sulfate to sulfide, which can be reoxidized or used by microbes. So, you have in certain cases biological and abiological sulfur oxidation, and then in others you have anoxygenic photosynthesis, and then once these are converted to sulfate, this can easily go to plants or absorbed by plants. Apart from these, we have many other geological biogeochemical cycles. Because of time constraints for this particular lecture, we will be discussing them in another module in this lecture series. Let us now discuss about the use of microbes as biofertilizers, and we will also discuss about the various class and types, and also a little bit about arbuscular mycorrhizal fungi as biofertilizer and certain endophytic fungi as biofertilizer. Biofertilizers or microbial inoculants are microorganisms that enhance plant growth and yield by colonizing the rhizosphere and plant roots. They improve soil fertility, protect against pests, and support stronger root development. Common biofertilizers include nitrogen fixing bacteria, phosphate solubilizing bacteria and fungi. These microbes provide essential nutrients and growth promoting substances, contributing soil health and sustainable crop production. So, what are the benefits of using biofertilizers? They enhance nutrient availability, improve soil health and causes less environmental pollution, and they are very important for sustainable agricultural production, enhances biotic and abiotic stress tolerance. They improve structure and function of our ecosystem. They are easy to use, promotes phytohormones production, and overall they are comparatively low cost and also can be produced at the farm level. So, let us look into the various types of biofertilizers. So, biofertilizers produced by heterotrophic bacteria or phototrophic organisms and fungi. So, in heterotrophic bacteria we may have nitrogen fixers or phosphate solubilizers, then we have SAC deaminase producing bacteria, and then zinc solubilizers. In phototrophic we have cyanobacteria, which may be symbiotic and associative, and then we have under fungi phosphate solubilizing and mobilizing bacteria, and also we have enriched compost biofertilizers. Nitrogen fixers may be symbiotic, associative, non-symbiotic. For example, we have Rhizobium, Azospirillum and Azotobacter respectively. So, if you look into the various mechanisms of various biofertilizers, in nitrogen fixing we can see that increase in soil nitrogen content happens by fixing atmospheric nitrogen and making it available to plants, and as already discussed, they may be freely being symbiotic or associative symbiotic, and these are the examples for these various types. Then we have phosphorus solubilizing ones, which solubilize the insoluble forms of phosphorus in the soil into soluble forms by secreting organic acids and lowering soil pH to dissolve bound phosphates, and this can be carried out by both bacteria and fungi like Bacillus and then Penicillium respectively. We have phosphate mobilizing biofertilizers. These transfer phosphorus from the soil to the root cortex, and these are broad spectrum biofertilizers. For example, you have mycorrhiza, arbuscular mycorrhiza and globus species. Then we have potassium solubilizing biofertilizers. These solubilize potassium silicates by producing organic acids that decompose silicates and help in the removal of metal ions and make it available to plants, and they may be bacteria and fungi like Bacillus and Aspergillus niger respectively. Then we have potassium mobilizing biofertilizers. They mobilize the inaccessible forms of potassium in the soil, maybe bacteria like Bacillus and fungi like Aspergillus niger. And then there are certain micronutrient providing biofertilizers which oxidizes sulfur to sulfates, which are usable by plants, or solubilize the zinc by proton chelated ligands, acidification and oxidoreductive systems. And these may be Mycorrhiza, Pseudomonas and Bacillus species. Then there are certain biofertilizers which provide plant growth promoters or hormones that promote root growth, improve nutrient availability and improve crop yield. So, we call them as plant growth promoting rhizobacteria, for example, Pseudomonas and Agrobacterium species. Let us now discuss about biofertilizers and their nitrogen fixing abilities. So, as already discussed, nitrogen fixers may be symbiotic organisms, or they may be free living or associative symbiotic, and also we have cyanobacteria based biofertilizers, and then there is Frankia based biofertilizers. In the case of symbiotic nitrogen fixers, microbes like Rhizobium leguminosarum form symbiotic relationship with leguminous plants and fixes atmospheric nitrogen in root nodules and provide the essential nutrients. Then we have the free living nitrogen fixers like Azotobacter, Crococcum, which is a free living bacterium that fixes nitrogen in non-leguminous plants like cotton and vegetables, reducing the need for chemical fertilizers. Then we have associative symbiotic nitrogen fixers like Azospirillum based Azospirillum basilense and other species associated with grasses to fix nitrogen and promote plant growth, fixing approximately 20 to 40 kg per hectare per year in land leguminous plants. And then we have cyanobacteria based biofertilizers, basically photosynthetic bacteria that fix atmospheric nitrogen in aquatic and semi-aquatic environments, as in the case of paddy fields, for example, Anabina and Nostoc. And then we have Frankia based biofertilizers. Frankia is a genus of nitrogen fixing bacteria that forms symbiotic relationship with actinorhizal plants, similar to Rhizobium's role with legumes. It helps initiate root nodule formation and fixes atmospheric nitrogen, like for example, Frankia enlii. Then we have phosphate mobilizing microbes, microorganisms that mobilize native and added insoluble phosphorus sources, including organic phosphorus through enzymatic hydrolysis, are termed as phosphate mobilizing microorganisms. The phosphate solubilizing microorganisms are also helpful in facilitating phosphate translocation and are thus included as PMM or referred to as PMM. Microbial phosphorus solubilization involves the organic acid theory with two mechanisms, namely the pH lowering, which directly dissolves organic mineral phosphorus, and then there is the organic acid anion releasing theory, which focuses on the ligand exchange. So, PMM may directly mobilize both inorganic and organic phosphorus. Indirect mobilization also of phosphorus can also occur. In the case of organic acid production, microorganisms produce organic acids like gluconic acid that lower the pH, dissolving inorganic phosphates. Then there is chelation, where chelatic agents such as 2-keto-gluconic, humic and fulvic acids bind to metal ions like calcium, iron and aluminum, aiding phosphate solubilization. Then there is inorganic acid production. Acids like sulfuric and nitric acids are produced by nitrifying and sulfur oxidizing bacteria, though less effective than organic acids for phosphate solubilization. Then there is proton extrusion, where phosphates can be solubilized by proton extrusion, lowering the pH without organic acid production influenced by nitrogen sources. And then there is exopolysaccharide production, where microbial exopolysaccharides, which bind to metals in the soil, indirectly aid phosphate solubilization alongside organic acids. Then there is siderophore production, where siderophores, which chelate iron, can enhance the solubility of iron bound phosphates. And there is also enzymatic actions of enzymes like phosphatases, phytase, and then phosphonatase, hydrolyze organic phosphates, releasing phosphate ions for plant uptake. Let us now discuss about potassium solubilizing and mobilizing biofertilizers. Potassium is the most important plant nutrient after nitrogen and phosphorus. It is abundant in soil, but only 1 to 2% is available to plants, requiring continuous replenishments for plant growth. Deficiency of potassium leads to slow growth, poorly developed roots, and low yields. Various bacterial and fungal strains employ mechanisms like acid production, chelation, and complexolysis to solubilize insoluble potassium into soluble forms. Examples of potassium solubilizing biofertilizers are Bacillus species and Aspergillus niger, which improve solubilization and mobilization. So, we have fungi and bacteria which can solubilize potassium, and there are different mechanisms. So, one is the PGPR attributes, where there is hormone production, biological nitrogen fixation, phosphorus solubilization, and root colonization and micronutrient solubilization. Then there is the biotic and abiotic stress tolerance induction, where SEC deaminase activity, antibiotics production, and chitinase activity takes place, and hydrogen cyanide production and siderophore production takes place. And there is certain mechanisms to improve potassium availability, like organic acid production, chelation cations bound to potassium silicate, and then polysaccharide secretion, biofilm formation, etc. So, one of the mechanisms of potassium solubilization is to release potassium nutrients by the production of different organic acids, which acidify the rhizosphere and enhance potassium solubilization from minerals. So, organic acids produced by potassium solubilizes different kind of sources and make the potassium available. So, these organisms we can call as potassium solubilizing microbes, or KSMs. So, the various acids may be oxalic, gluconic, 2-ketogluconic acid. Some produce lactic, others produce malic, acetic, tartaric, fumaric, glycolic, malonic, formic, citric, succinic, and propionic acids. Low pH due to the release of these acids facilitates the production of organic acids and protons, which directly dissolve potassium minerals, releasing potassium ions via protonation. These organic acids can also release potassium ions from potassium bearing minerals by chelating aluminum, silicon, calcium, and ferrous ions, disrupting the lattice structure and releasing potassium into soil solution. The next important type of microbes are microbes which can solubilize zinc, or zinc solubilizing microbes. For example, zinc solubilizing bacteria efficiently convert complex inorganic zinc into available forms of plants, with examples like Pseudomonas fragi, Plantua dispersa, and Pantua agglomerans. Silicate solubilizing bacteria play a crucial role in solubilizing insoluble silicates, as well as potassium and phosphates, enhancing soil fertility and plant production. The silicate solubilizing bacteria and hinged biofertilizers like Bacillus species improve the photosynthetic efficiency of plants like Brassica jansia. So, if you look into the mechanism of zinc solubilization, so we have organic acid production, and then acidolysis, then there is chelation, and then there is exopolysaccharide secretion. All these will lower the soil pH, and then the insoluble zinc will be solubilized by the action of these different mechanisms, and then these are made available for the plants to absorb. And overall, these root will contain the zinc solubilizing bacteria, and due to availability the proliferation of these organisms will take place. Let us now discuss about arbuscular mycorrhizal fungi. Here in this diagram you can see the effects of arbuscular mycorrhizal fungi. So, here we have these mycorrhizal species over here. So, basically the AMF or arbuscular mycorrhizal fungi, here fungal and plant genotype plays an important role in climatic conditions as well as soil variables, and then mycorrhizosporic bacteria and anastomosing ability of the pesticides also play a critical role. So, we have certain species known as the mycorrhizal helper which influence in many of these important events. Then we have plant growth promoting bacteria which synergistically interact with these AMF. So, it is a little bit of complex interaction. As you can see, overall these AMF will lead to the growth and mineral nutrition improvements, and then secondary metabolism modifications, and also tolerance to biotic and abiotic stresses, as you can see in this diagram. So, and then overall they help in improving the soil biological fertility and the soil structure. So, these arbuscular mycorrhizal colonization has positive effects, extending the hyphal network beyond the phosphate depletion zone. So, you can see here the gray area which is the phosphate depletion zone, and then it helps in assessing a larger soil area for phosphate uptake. Mycorrhizal phosphate depletion zones, which are the purples one, form around the arbuscular mycorrhizal hyphae, as you can see in this picture, and shows improved assimilation of nutrients like nitrogen in the form of ammonium, and then also zinc. Benefits of these colonization include enhanced tolerance to abiotic stress through systematic acquired resistance induction. So, due to these two figures, you can see there are on the left side no colonization, and here there is arbuscular mycorrhizal colonization. In this case, here there is increased resistance to foliar pathogens, then increased drought tolerance, which is absent over here. Then it also has increased salt tolerance, not available in case of no colonization, and there is the systemic acquired responses or resistance in this particular case. So, overall arbuscular mycorrhiza are highly beneficial to overall plant growth. So, how do mycorrhiza establish? So, plant defenses signaling is modulated during mycorrhizal establishment in the following stages. Number 1, during germination, arbuscular mycorrhizal fungi grow towards the root and form appressoria. Okay. On the root surface, this stage, at this stage the plant responds with an increase in salicylic acid levels. In a compatible interaction, these salicylic acid levels decrease as the fungus colonizes the cortex. Then in this particular zone you can see that. Then there is the jasmonic acid biosynthesis, which takes place in arbuscule containing cells. When mycorrhiza forms, it prepares the plant's tissues to respond more quickly and effectively when an attacker strikes. This means that the plant can activate its defenses faster, which helps in resist damage better. Let us now discuss about endophytic fungi as biofertilizer. Endophytic fungi which live without plant tissues without causing harm significantly enhance plant growth and resilience. As in the case of Piriformospora indica, we know for its ability to promote nutrient uptake. This enhances the availability of essential nutrients such as nitrogen, phosphorus and potassium by improving root function, and it also helps increase biomass production and contributes to higher yields in various crops. And it accelerates early flowering and seed production. It stimulates hormonal activity that supports lowering and seed development. Additionally, P. indica helps plants to tolerate abiotic stress, including drought and heavy metal toxicity, and boosts resistance to biotic stresses from pathogens and herbivores. So, we can see here the endophytes which we can use as biofertilizer. So, it can have direct effect or it can have indirect effect. In the case of indirect effect, it helps in nutrition acquisition on plant growth and development, and in the indirect effect they also help as biocontrol agents. And then from the application point of view, they release bioactive compounds which can be having importance in agriculture, and they can be of medicinal value and as well as in certain other industrial applications. So, leaving these two aside, overall the endophytes directly and indirectly help in enhancing stress tolerance and disease resistance in plants. Then they promote bioactive substance accumulation and increases plant growth. So, here we can see the spores of Piriformospora indica colonizing the roots, and overall they increase in the nutrient acquisition, which helps in many of these outcomes, and they improve rhizosporic microorganism activity. So, these are the positive effects of Piriformospora indica on overall plant growth as an endophyte. Then we have other endophytes which are used in agriculture. For example, we have Clavicipitaceous endophytes. So, particularly Apiclo species are mainly used for biocontrol in pasture grasses. These Apiclo Cynophila enhances pest tolerance in tall fescue by producing alkaloids that deter herbivores. Then we have ryegrass varieties which have many of these Clavicipitaceous endophytes that exhibit reduced damage from insect herbivores, and in countries like New Zealand and Australia. However, their application has been largely restricted to grasses, limiting broader use in other crops. Endophytic fungi also helps the plants in overcoming cold stress effects, or helps in cold stress resistance. For example, these very species Piriformospora indica help plants withstand low temperatures by enhancing ROS scavenging. This improves the plant's ability to detoxify reactive oxygen species, reducing cellular damage, and then boosting photosynthetic efficiency, ensures continuous energy production and metabolism in cold conditions. Increasing protective enzyme activity, this elevates levels of enzymes that neutralize ROS, protecting against oxidative stress, and promoting osmoprotectant accumulation, which increases soluble sugars and amino acids that stabilize cellular structures under stress. And it stimulates cold responsive genes, enhances the expression of genes involved in cold acclimation, and then improving resilience as seen in crops like bananas. So, with these we come to the end of this lecture. Thank you for your patient hearing.